Thanks so much, Dr. Hernandez. I really appreciate that. Nice intro. So I'm going to be talking about sex differences today, and specifically risk, expression, and treatment for Alzheimer's disease. I'll start with just a very brief overview, since this is a very well-informed audience, of what Alzheimer's is and how I contextualize it in my research. I'm then going to talk about symptoms or pathology of Alzheimer's disease and whether or not they're expressed differently in men and women. I'll then talk about sex differences and risk factors, and then ask whether sex-specific treatment strategies might help individuals reduce their risk of cognitive decline. So I just wanted to note that I use the term sex in a very oversimplified, dichotomized way. And sometimes I use it to incorporate gender factors. I realize that this is an oversimplification and that it's a lot more nuanced than that. So what is Alzheimer's disease? It's a brain disease, of course, and it's the most common cause of dementia. And dementia, in turn, is the progressive loss of cognitive abilities and ultimately independence due to brain disease. And the definition of Alzheimer's disease is getting more and more complicated. So I did want to give this sort of operational definition today. 1 in 3 of our seniors dies with Alzheimer's or another form of brain disease causing dementia. And so it's horribly, horribly common. I'm sure that everyone on the talk today has been impacted in some way by it. This slide is about a year or two out of date. There are now more than 7 million Americans living with diagnosed Alzheimer's disease and many, many more with undiagnosed Alzheimer's. Nearly 2/3 of those are women. And that is not just because women live longer than men. There are other factors that go into that sex disparity. What does Alzheimer's disease actually do in the brain? Well, as we know, amyloid plaques build up in between the neurons very early in the process, and they don't seem to cause too much trouble unless tau pathology builds up in the form of neurofibrillary tangles within the cells, which correlate very strongly with synaptic loss and ultimately atrophy. But we know more and more that beyond amyloid and tau, the brain's immune and inflammatory response is involved in Alzheimer's disease. So it's a complicated relationship that we'll touch on a little bit further, but the microglial and astrocytic activation is an important component of how the disease spreads in the brain. There's a specific timing of changes that has become to be pretty widely accepted, that amyloid spreads very early in the disease, potentially about 15 years before we see clinical symptoms in the form of memory loss. We can visualize that on amyloid PET or on CSF. About 15 years after that widespread amyloid deposition, we start to see problematic tau spread. And then that is initiated around the same time as neurodegeneration, with neural changes being evident with FDG PET, sort of metabolic changes, fMRI changes earlier than structural changes. But ultimately, we have this cascade that we see of biomarker changes with clinical changes going along with it. And there's this preclinical phase, which offers a really great opportunity for intervention, where we know somebody is on the cascade, but ultimately don't have at that point any cognitive dysfunction. And then we have this prodromal or MCI phase, which is also really important. I'm going to talk about that a lot today as well. I'm very interested in tau pathology, in part because I'm a neuropsychologist and it correlates so strongly with cognitive change. And what do I mean by tau pathology? So in the background of this slide, you can see on the left some really nice healthy cells, which are presumably being helped along by these stable microtubules, which is what we think tau's role is in the healthy brain. But once we start to see dysfunction, we see oligomerization building up in the cells, and we think that's how tau spreads cell to cell. This builds up in cell bodies as intracellular neurofibrillary tangles, which prevents the normal functioning of the cells. So this is where we see with enough of these tangles, we can pick it up on tau PET. And ultimately, the neurite dies, leaving only the extracellular tangle or ghost tangle. Tau spreads in the brain in standard late onset Alzheimer's disease with an amnestic profile. We can see very predictable spread of tau in the brain. So in stages 1 and 2, it's very much contained within the locus coeruleus entorhinal cortex. And then stages 3 through 4, it starts to become more widespread in regions that are important in memory and language and executive functioning. And generally, this lines up with what we see clinically as mild cognitive impairment. In stages 5 through 6, the tau is very widespread throughout the brain, and that sort of lines up with dementia. It's not a one-to-one connection, but in general, we do see a lot of correlation between where the tau is and how much tau there is and what the clinical symptoms are and how severe they are. So what about neuroinflammation? And I want to mention this at a very high level. There's a lot of very complicated science here that many people on the line today spend their lives working with, could explain this much better than me. But I think what we're seeing at a very high level is when the pathology starts to spread in the brain, there is a response from the microglia and the astrocytes and this sort of glial activation, which initially might be protective, but ultimately becomes problematic. And we get this sort of detrimental loop where the glial activation is actually promoting the spread of tau. So inflammation might be something, neural inflammation might be something that we can target again with different interventions to help stop the spread of tau in the brain. Treatments so far in Alzheimer's disease haven't really worked yet. So on the left here, this is the Alzheimer's disease drug development pipeline, which is put together every year by Jeff Cummings. And you can see this is the 22 one, but there's a 23 and I think even now a 24 one, where we can see the different slices of the pie chart represent the different kinds of medication that are being developed, from disease-modifying biologics, small molecules, neuropsychiatric symptoms. Dr. Cummings has a particular interest in that, cognitive enhancers. And you can see there's a huge number of drugs currently under development at different phases of development. And these are really, really expensive. They cost billions of dollars and they take years to try to develop a drug. And ultimately, so far, we've had more than a 99% failure rate with drug development in Alzheimer's disease, which is pretty bleak. There is definitely some hope on the horizon with recent new medications being approved by the FDA that we'll talk about today a little bit. But there's another kind of hope that I think is really exciting with really new understanding of modifiable risk factors. So on the right side of this graph, you see a summary from the Lancet Commission, the more recent paper from 2024, where they've put together the evidence for modifiable risk factors that impact at early life, midlife, and late life. And some of these are more helpful when we're thinking about changes in policy at a more sort of national level, but other things like changes to hearing loss, cholesterol, depression, and late life risk factors such as social isolation and visual loss are really important because those are things that individuals can target. And you'll see that there's a lot of vascular risk factors involved there: diabetes, smoking, hypertension, obesity. And ultimately, all of these different risk factors combined explain about 45% of dementia risk, according to the authors. They did not sex stratify their analysis, sadly. I wish they would. But ultimately, I would like to ask the question today whether sex stratification could help us find more effective treatments. So I wanted to touch on one of the recent FDA-approved medications, just at a high level. just to explain what I mean when I say that the drugs haven't really worked yet. So this is a drug that's exciting. It can take amyloid out of the brain, which is really cool. And it does slow progression of the disease. This is in people with early dementia, so MCI/early dementia. And over 18 months, we see this attenuation of decline. And this translates to about a 4 to 6 months prevention of decline in individuals. So it is something, but it does come at a cost with risk factors and the need for multiple brain scans and other costly ventures. And there are a lot of cons in addition to some pros. So definite hope, but maybe not super exciting for most people with Alzheimer's disease. So that was the background, just to set the stage for what I'm going to talk about today. So I'm going to divide my talk into 3 sections, and I'm going to start with sex differences in the expression of Alzheimer's disease. So Alzheimer's disease doesn't just occur on a blank canvas. It occurs on humans who have a lifespan of cognitive functioning behind that. And overall, throughout our lives, men have a slight visuospatial advantage. So on the graphs on the left there, you can see points from adolescence, young adulthood, and older adulthood. And you can see men in the heavy line there do a little bit better on visual tests, visuospatial tests in general, and visual episodic memory, more particularly at the bottom, than women in the dotted line. So that there is this slight difference on the memory tests. Those error bars are overlapping. It's not really significant, but on general visuospatial ability, there is some difference on average. For verbal ability on the right-hand side, women have the advantage. So women, again, in the dotted line are higher up here, and there's nice separation between the error bars throughout life, between the men and the women on verbal skills in general at the top, and verbal episodic memory at the bottom. So women are coming into old age with a verbal advantage compared with men. And this is exacerbated a little bit by the kinds of tests that we generally use to assess memory in trials and studies of Alzheimer's disease. So this is an example of the California Verbal Learning Test. The actual words have been switched out, but this is a kind of standard way that we assess memory in humans, where we'll read them a nice long list of words, and then we'll ask them how many they remember, and then we'll keep reading that list until they've remembered as many as they probably can over 3 trials, or in this case, 5 trials. And then after a delay, we'll come back and ask them how many words they recall, and then maybe a recognition trial as well. So this is obviously really tapping into verbal memory abilities, and women tend to do a lot better on these tasks. And that's reflected. This is work by my colleague, Erin Sunderman, who's listening today. Hi, Erin. So Erin did this work a few years ago showing that despite higher levels of amyloid on the x-axis here, women tend to do better on a test very similar to the one I just showed you. So this is the Ray Auditory Verbal Learning Test. She's got the women in blue and the men in the sort of salmon-y color here. And you can see that in the overall sample in the top left, this is data from the Alzheimer's Disease Neuroimaging Initiative. Women tend to be in the higher levels, and the mean there is in the dotted line. And that's true when she breaks up the sample by normals in the top right, amnestic, MCI on the bottom left. So they're still maintaining, the women are still maintaining that advantage over the men. And then we see a loss of that advantage at the dementia phase, where the lines begin to connect at the end. So throughout normal cognition and the amnestic MCI, we do see this verbal memory advantage in women despite similar levels of pathology. And this is a problem because when we look at men and women in studies, we're probably not looking at similar levels of pathology. So basically, women have this high verbal cognitive reserve, as if they're this heavy line at the top of this graph on a verbal memory test score, and men have a lower reserve going in. So at any one particular visit, we're going to see that men will have less tau or other pathologies, but particularly tau, and women will have more tau. So we're comparing apples and oranges when we have men and women based on a verbal memory cutoff score. We could circumvent that a little bit by using different composite scales weighted for the skills of men and women in general. So this is a study that I did with Steve Edlund a few years ago and his team, looking at what would happen if we created sex-specific composites, again, from the Alzheimer's Disease Neuroimaging Initiative data, to create different sets of tests and weightings of tests based on how where women's skills are and where men's skills are. And this graph will show us how many people we would need in a 2-arm clinical trial if we use these composites as outcomes instead of standard composites like the PAC in the solid line there. So for a power of 0.8, we would need about 250 men or women in the study. Whereas if you follow the trajectory of this heavy line out, you would see it would take many, many hundreds more before we got to 0.8. So when we think about the cost of studies and the tendency for these studies to be negative clinical trials in Alzheimer's disease, one thing that we might think about is what are the outcomes and who are they best applied to. So there's these sex differences in cognition in terms of expression of Alzheimer's disease, but there's also sex differences in pathology more specifically. So women accumulate more tau than do men, and we can see that on this PET study that we did with the ADNI data again a few years ago, where we can see that women have more tau than do men, and that's shown here with the more yellow coloring compared to red. The distribution is very similar, with a lot of tau in the medial temporal, not medial, but the temporal regions here, and more medial temporal down here. But there's just more of it in women. These are women with MCI who are amyloid positive. So these are definitely on that Alzheimer's trajectory. And this is seen as well in other studies with CSF, autopsy. This is definitely not one study that's showing this. It's a pretty widely accepted difference at this point. And when we combine the additional tau deposition with the verbal memory advantage, we see that women overall appear to be able to bear this bigger burden, where this is Dino Digma when he was in Jim Brewer's lab and my lab a few years ago. He's now on his neurology fellowship in Stanford, I believe. And he showed that using residuals, that women have this cognitive advantage over men. So women here are in red, men are in blue. And we can see that with more tau, women do better on verbal memory and have more of the dots above this heavy line, showing that they have this stronger reserve. This is with PET. This is just another way of showing the same thing. The women have the higher distribution towards the top. And then he also looked at post-mortem path analysis in the NAC data set. So this is in autopsy. Women are showing a similar thing, where if they have BRCA and tau in BRCA stages 1 and 2, so very early changes, they're definitely doing a lot better than other men on this verbal memory test before death. And at 3-4, they're maintaining quite a bit of that advantage. But then just like in Dr. Sunderman's work that I showed earlier, once we get to the end stages, in this case, BRCA stages 5 and 6, we no longer see a verbal memory advantage for women. Everyone's kind of doing badly. So I mentioned earlier when I showed this graph before that the preclinical phase is particularly exciting, where we see an amyloid PET positivity or amyloid positivity and some other biomarker, but there's not a strong memory problem. However, people think that there might be some subtle cognitive changes at that stage. And this anti-amyloid treatment in asymptomatic Alzheimer's, spelled wrong, study, the A4 study, is a really interesting kind of treasure trove of data. We looked at the baseline data with Xin Wang on the team. And one of the reasons why the A4 is really exciting is, well, one of the reasons is they're looking at all preclinical Alzheimer's disease. And another one is the cognitive measures that they used. So they used things like the face name association test and different cog state measures of cognitive functioning, which are computerized measures, so that they're using sort of out of the box tests. And what we showed in our analysis, I'll show you the graphs on the next slide, it's that some of their cognitive tests are significantly related to the level of tau in the brain, shown with tau PET, but only in women. And this could be really important because more and more our intervention studies are targeting those preclinical periods of the disease, and we want good outcome measures that are actually going to show changes over time. So these are some of the graphs, and I just blocked out the ones that weren't significant so we could look at the ones that were. And if we look at something like the PAC composite measure, what you see is that the women who are in red here show a stronger association with the amount of tau PET and their cognitive score than do men. And actually for men, it was not significant. And that's the same pattern that we see on multiple of these different measures that they used. None of them were reversed. So if I showed you the other ones, you wouldn't see the reverse pattern where some tests were significant for men. They were either not significant for either or significant for women. Here's the face name learning test. So for many of these, we see these strong relationships in women and a much flatter relationship with men. And I think this really highlights an opportunity where we could be learning about early changes in cognition in Alzheimer's disease, but only if we stratify by sex. So to summarize this first section of my talk, where I've asked, are symptoms or pathology of Alzheimer's disease expressed differently in men and women? I would say yes, on multiple fronts. So women have this surge of tau pathology during the MCI phase. So there's a definite difference in pathology. And then women have this lifelong verbal cognitive reserve, which obscures their early decline. But with the right tests, women's early cognitive decline in relation to tau pathology is quite evident. So definitely differences in this expression of Alzheimer's disease. So for the second component of the talk today, I wanted to talk about sex differences in risk factors for Alzheimer's disease. And I'm going to start with mice. So this is a mouse who's training for his Maurice Watermays debut in nice clear water. Of course, Maurice Watermays has obscure water, but that wouldn't be such a cute picture. So this study, oh, I haven't put the reference in. No, I have, by Gannon and colleagues, is a really interesting one because they took different mouse models of Alzheimer's disease and mixed dementia, as well as wild type, and they fed them either a low-fat or a high-fat diet. And what they saw is that female mice who have a high-fat diet do worse on the Maurice Watermays probe trial, which is a measure of memory. And that's true whether they have the Alzheimer's disease variant or a mixed dementia variant. They're doing worse than their fellow mice who have a low-fat diet. Whereas in male mice, we don't see that difference. There were no sex differences, and I'm sorry, there were no diet differences whatsoever. So that's an interesting piece of information in terms of diet and how it might impact cognitive outcomes in either sort of wild type mice or those who are destined to get dementia. But what about humans? So pre-diabetes is incredibly common, and it's the impact of poor diet. It's impacting 1 in 3 Americans right now. And interestingly, pre-diabetes is associated with an increased dementia risk in women, but not men. So that's been shown very nicely again by Dr. Sunderman in this work where she was looking at the Alzheimer's Disease Neuroimaging Initiative, and she looked at pre-diabetes status, which is in the dotted lines here, with women in the green and men in the blue. And what she saw was that men and women with pre-diabetes status do a little bit worse in terms of brain metabolism, and there's no sex difference there. But there is a sex difference in cognition, where women with pre-diabetes do much worse on executive and language skills than do men with a similar level of prediabetes. So this is an example of the executive skills that we're showing here, where the patient or participant has to join the numbers and letters in alternating order, so going from 1 to A to 2 to B, and so on. So it takes quite a lot of skill, and we're showing that prediabetes impacts that kind of cognitive skill particularly. This is another study by Shen Wang on my team, going back to the A4 data set, so these preclinical individuals. And this is showing that the relationship between things that we think of as bad vascular risk factors, like being overweight or obese, are complicated when we look at older women and men. So in older women specifically, we see an advantage of being overweight or obese compared to being normal weight in terms of entorhinal tau. We see that in women specifically. We do not see a similar relationship in men. And this has been documented elsewhere, where the being a little heavier for an older woman appears to be associated with less Alzheimer's risk. It's also associated with better memory. So you see here, this is a forced choice recognition test. And what we see is the women who are obese in black or overweight in dark gray are doing better on that test compared with women who are normal weight. For men, there's a suggestion of a flipped relationship, where on the mini mental status examination, which is a cognitive screener, obese men are doing a little bit worse than our normal weight men. But we don't see it in the more nuanced cognitive tests. So this is suggesting that, yes, vascular risk, when we look at diet and prediabetes, is bad, especially for women. But there's a lot more nuance when we look at something like body mass index, where a higher weight might be suggestive of some kind of protective effects. I wanted to mention sex, gender, and sleep apnea as well. And I'm bringing up gender here because we see sleep apnea as being a big risk factor for Alzheimer's disease. But women are often not diagnosed with sleep apnea, whereas men are. And that's in part because women have a different presentation to men. They have, in general, less loud snoring, less witnessed apnea, so the sort of gasps and snorts that we associated with suddenly gasping for breath in the middle of the night. Women are more likely to present with headaches and complaints of unsatisfying sleep, reflecting undiagnosed sleep apnea. And in older papers, it used to be reported that women only made up 10% of cases. So it was very much seen as a man's disease. 30% is considered to be a more accurate percentage of women represented in the cases overall throughout life. But if we look just at women after menopause age and compare them with similarly aged men, there's actually no difference in the incidence and prevalence of sleep apnea. And this is important because it's highly treatable. And I wanted just to plant that little seed for something I'm going to come back to in a little while about sleep apnea. And sleep apnea, for those who might not know, is when there's interruptions due to obstructions of the airway during the night, and that causes less oxygen to get to the brain, which is really relevant for structures like the hippocampus, which are some of the most sort of oxygen-hungry parts of the brain, and also really relevant in memory and Alzheimer's disease. So Erin Sunderman and I've mentioned several of her studies, and she and I have done similar work for many, many years. And then when we were both working at UCSD, we were having coffee and talking about how we might collaborate. And we were both thinking, this was about 5, 6 years ago, that inflammation might be a really important mechanism to tie in some of these risk factors with pathology and cognition. And this is work by Dr. Rachel Bernier, who ended up being a postdoc in our lab, where she's showing that a particular inflammatory marker, STNFR2, is related to cognition with the MMSE, and actually it's also related to tau in women, but not men. And this relationship is actually mediated by tau in CSF. So what we're seeing here is that inflammation might have some kind of link between cognition and tau. And we know that poor diet, insufficient exercise, and poor sleep all are related to an uptick in inflammation. So that became part of our thinking and led to a study that we now co-lead, which I'll introduce to you later. I wanted to mention a non-modifiable risk factor, genetics. And this is Amaryllis Tasignia, who was in our lab and is now at USC getting her PhD. But she looked again at the Alzheimer's Disease Neuroimaging Initiative. And this is a pretty healthy group of participants in general, but there is some range of cardiovascular risk. So she calculated the Framingham cardiovascular risk score on everyone, which brings together several vascular risk factors, including cholesterol, diabetes, blood pressure, etc. And she looked at scores on the Framingham risk score and also tau in different regions important for Alzheimer's disease. And what she saw is that there was a specific relationship in people with ApoE4, where women with an ApoE4, one or more ApoE4 alleles, show this very strong relationship between vascular risk and tau, whereas men do not show a similar relationship. And this is interesting because, again, these individuals are quite healthy. There's not a ton of vascular risk. So this is suggesting that for a woman with an E4 allele, having even a little bit of risk might be really problematic. This is another study I just wanted to highlight by, again, Xin Wang. And this came as a follow-up for an earlier study led by Junche Fan that we did looking at a polygenic hazard score specifically for men or women. So we then wanted to see if some of the specific genetic variants that were not ApoE4 that were related to Alzheimer's risk in women and not men might be related to tau. So that's exactly what Xin did. And I just wanted to highlight some of these results. So what we have here are graphs showing the amount of tau distribution in carriers and non-carriers who are men and women, and the amount of tau in BRCA region 1 on the left, region 3, 4 in the middle, and 5, 6 on the right. And what Xin found is that there are indeed sex differences in how much these different genotypes explain tau, but they're really more specific to BRCA 3, 4, which is where we see the most tau spread during MCI. which is super interesting because we see that surge in MCI for women and not men during that period. And so there's these specific genetic risk factors that are potentially drivers of that, that association in women and not men. And this is again beyond ApoE4. So to summarize the risk factors, well, we had expression, so we know that there are cognitive and pathological differences in terms of men and women. And there's also these differences in risk, whether we're looking at diet, cardiovascular risk factors, sleep apnea recognition and diagnosis, and genetic risk factors interacting with modifiable risk. And potentially inflammation could be a common theme underlying a lot of this. So how do we capture this in a study of Alzheimer's disease? So I mentioned earlier that Erin Sunderman and I were interested in inflammation and tau, and that she and I decided to study women in isolation. We felt like there were enough differences in risk factors and expression to justify just looking at women for now, and then in the future, hopefully, we will expand to look at men. But we came up with the Women Inflammation and Tau study, which I'm going to show you a little bit about now. We're very excited about this study. We're looking at women who are at least 65, who have no formal dementia diagnosis, and who are at heightened risk for Alzheimer's disease due to some combination of the following factors. So their demographics, specifically age, genetics, and/or family history. And the women in our study are pretty amazing. They go through a quick screening call, and then if they are eligible and they agree, they come in for a lumbar puncture, blood draw. They go home with sleep and activity devices. They come back for another visit where they have cognitive testing and MRI scan, and a third visit where they have a tau PET scan. And then 2 years later, they have some of that repeated, not the lumbar puncture, thankfully. And this is what we're trying to understand. We would like to know if these modifiable risk factors, so we've focused in on sleep apnea, exercise, and insulin resistance or sort of prediabetes, impact Alzheimer's disease outcomes. So we're interested, of course, in cognition and also pathological tau. And we use this very sensitive PET tracer to look at that. This is the tracer that we're using, MK6240, compared with flutalsapine, which is the FDA-approved one, which is a bit less sensitive. And we think that these pro-inflammatory cytokines are potentially mediating some of this relationship. And we're also looking at sex hormones, including testosterone, which we have some really exciting data on, which I don't have time to show today. The cognitive testing in our study is, of course, not just verbal. We've sort of learned our lesson that verbal memory is not sufficient or not sensitive enough to detect some changes for women. So we do verbal memory. We do the same battery as the ADNI study that we have learned so much from, but we also focus on nonverbal memory. So this is an example of stimuli that we might show someone for 10 seconds, and then we take the page away and ask them to draw the shapes in the same location that they saw them on the page. And similarly to the word list that I showed you earlier, we repeat the learning trial several times to give them an opportunity to learn as much as they can. And then later on, we do a delayed recognition. I'm going to show you some of the very early results. So this was a preliminary study by our first project coordinator, Alex Shepard, and she looked at tau PET SUVR in the metatemporal region, along with percent correct on delayed recall on the visual test in blue and the verbal test in green. And as you can see in our women, that we see much stronger relationships for the visual test compared with the verbal test. And this is just a different brain region, but with a very similar pattern. I also highlighted sleep apnea earlier, and that's graduate student Kitty Liu's area of expertise. So Kitty has been working on this, and I think Erin and I have been particularly surprised that so many women in our study have sleep apnea. Very, very few, maybe one or two coming into the study so far out of the 50 plus that we've tested, were diagnosed with sleep apnea or were concerned about sleep apnea. They all have a sleep test as part of the study, a home sleep test, and about 70% of the women so far have had positive sleep apnea tests, and about half of those have had moderate to severe symptoms. What's more, there's a really strong relationship between sleep apnea symptoms and tau in our data. But what I'm showing you here isn't even from the sleep test, it's just from a sleep complaints questionnaire. And Kitty looked at the Pittsburgh Sleep Quality Inventory, which is just a 10-minute sort of questionnaire about how satisfied someone is with their sleep. And she looked at that in comparison to the tau PET in the top left. As you can see, there's this really strong relationship just between sleep complaints and tau, and then memory. And you can see the visual test in green there, showing a very strong relationship, whereas the verbal tests are a little bit more noisy, as we'd expect with a verbal memory advantage. And on the bottom there, you see the relationship between tau on the x-axis and memory on the y-axis. So sleep is really important for our women anyway. And we see that when we, especially when we don't use a verbal memory test, but instead a visual memory test. Another study that's on sleep has come from postdoc Dr. Jordan Stiver. He took a slightly different angle, and he looked at some of the actigraphy devices to look at variability in sleep efficiency. And what he sees is that having variable sleep, so more sleep some nights, less sleep other nights, is problematic, but specifically for those women in green here who have one or more E4 alleles. And he sees that whether he looks at metatemporal tau again, or the verbal memory test, where this variable sleep is problematic. So we're big on sleep in our lab. So WITS requires a lot of visits and some more invasive testing with the lumbar puncture. And a couple of years ago, Dr. Sunderman and I were talking about how we had a very non-diverse data set and what could we do to correct that. And as part of that conversation, we figured out that we really should be honing in on some of these relationships in the most impacted group in America, which is Black women. So instead of trying to diversify WITS, we came up with an entirely new sister study that's devoted to Black African American women. So, you know, we've heard already that 2/3 of all Americans with Alzheimer's and related disorders are women. But Black African Americans are more than twice as likely to receive that diagnosis compared with whites. And as a result, they're the most sort of at-risk group for women who are Black or African American are the most at-risk group. And those slides were beautifully made by Joy Stratford in our lab. So we came up with this study, the BWIT study, which we're doing in collaboration with UCLA. Here at UCSD, we collect all of our data from community sites, so churches and community centers, and we're able to leverage the amazing advances in blood-based markers of both inflammation and tau in recent years. So we're looking at 100 women overall, and they have similar inclusion criteria to WITS. And similarly, they have a 24-month follow-up, but these 2 visits are done in person in the community. So I just wanted to mention that study. We don't have data to share just yet because we're in the recruitment phase, which is going very well, thanks to a really incredible team of people who know what they're doing, who are advising us and working with us. So I wanted to summarize quickly the second part of my talk. So do risk factors differ in men and women? So yes, they do, is the short answer. Whether we look at modifiable risk factors such as diet and vascular risk, genetic risk factors, and interactions between these modifiable risk factors such as sleep or vascular risk and genetic risk. So we felt that these complexities and sex differences really justified a focus on women as we do in WITS and BWITS. But we also want to make sure that we focus on those who are most at risk, which is why we look at BWITS, and results will hopefully be ready to share in a year or two there, if I'm invited back. So the final component of the talk today, is not that one, it's treatment. So why do sex differences matter? So I wanted to show you these results that were shared at CTAD a couple of years ago, right before lecanemab received its FDA approval. And they had about 50% women and men in the lecanemab study. So there were many hundreds of women, well over 800, and a little over 800 men in the study. And on these forest plots, what you want is your error bar to not touch the 0 line. If it touches the 0 line, it's a non-significant finding. So what happened is overall, we see that the results, whether we look at the ADAS-Cog14, which is a cognitive test, or the ADCS-MCI-ADL, which looks at activities of daily living, overall, the results favored lecanemab, and they were significant. They were not touching the 0 line. But when we look at women and men separately, we see for women, the bar does touch the central line, suggesting that these results were not significant. And this was not something that they powered for as a preliminary outcome. So they went ahead and still got the approval. But for men, the results seem to be much better for this particular drug. I suspect that's because of their inclusion criteria, which were based on verbal memory tests. So I suspect that the women in the study had more tau pathology in their brain and were more advanced pathologically than were the men, but we don't have those data to share. Donanemab might be slightly better. They used a different way of including people based on biomarkers and not cutoffs on verbal memory measures. So in this case, we see actually kind of a flip for this particular outcome where the men are not significant and the women are. But for the CDR sum of boxes, we see that there's an advantage for both men and women. So this, I think, shows how using sex and inclusion might be, sex and how we select our criteria for inclusion and outcomes might be really important. There have been a handful of studies looking at sex differences in other drugs over the years, but it's not a lot. So this is a study that was, I think it was back in sort of 2014 or 2015. I don't have the year on here, unfortunately. But they looked at 144 patients with Alzheimer's disease in Italy. 157 of those were receiving either donepezil or rivastigmine, which are cholinesterase inhibitors, and 27 with no treatments. They also identified 2 estrogen receptor genotypes, and the outcome was MMSE, so an overall cognitive screener. And what they found, and we can see in this table down here, is that the female patients differed from the male patients. They declined more slowly on the MMSE if they were on the either treatment, donepezil or rivastigmine, whereas for men, we don't see that advantage. And they also found that the results related to donepezil specifically were associated with the genotype for a specific estrogen receptor, which I think is really exciting. And it's sad that we haven't seen more follow-up on some of these studies. There was a paper that came out looking at multiple randomized controlled trials for Alzheimer's disease a few years ago that showed only 7 of these studies had reported a sex-stratified response. And indeed, the lecanemab data was presented at a meeting and available online, but it was not published, whereas the donanemab more positive data was published. So that's sex. I did want to touch a little bit on race as well, just to show why it's so important that we focus on race as a factor. So if we look at the numbers involved, again, this is the lecanemab study, we see that there were only 40 Black participants, whereas there were well over 800 white participants. And we also see that the results were all over the place, not surprisingly, given the small number for Black participants. So that's problematic, and it's similar for the Hispanic group right above. And donanemab, we see something similar, where for their non-white groups, Asian, they had 80, and Black African American, they had 30. And again, the results are quite different. So I think focusing on race will be really important so that we can understand who these drugs work for and treatments in general moving forward. I wanted to finish with a study of modifiable risk factor intervention. So this is a meta-analysis of studies on 45 adults 45 years and older, looking at cognitive outcomes and whether or not exercise is efficacious in improving those outcomes. And this is a study by Cindy Baha from University of British Columbia, I believe. And she looked at studies that had more than 71% women versus studies with less than or equal to 71% women. And she found that studies in general with exercise, but especially those with aerobic training, had this real advantage for women than did men. And that was specific to tests of, or not specific, but it was strongest in tests with executive functioning outcomes. So it was also seen with memory and processing speed, I believe, but strongest with executive functioning. So this shows that even with the modifiable risk factors, we're seeing sex differences with interventions. So overall, I wanted just to leave with some take-home points today. So men and women differ in how they express Alzheimer's disease cognitively and pathologically. Risk factors different by sex. Treatment effects likely often differ by sex. Sex matters in general. Please think about it in your research. And we need careful consideration and inclusion of non-white race groups, vital to make a difference with this disease where they are most common. So thank you very much. I wanted to say thank you. Feel free to email me, and thank you to the gazillions of people who help with all of this research. I'll leave that one up, but there are 2 slides of thank yous. So yeah.